Good afternoon, everybody. I hope my voice is audible. We'll just wait for 2-3 minutes for other people to join and then we'll start the session. Okay. Okay, so we'll start the class. So, good evening, everybody. Uh, in today's lecture, we'll be dealing with the questions which are related to week 4 of the NPTEL Introduction to Developmental Biology course. So, let us come to our first question. So, our first question is, match the following types of lead 60 alleles to their phenotype. Okay, so it is a match the following kind of a question. So, your options are antimorphic, complete loss of function, and hypermorphic. And your options are multivalva, valva, less, or lethal. Okay, so this is related to the vulva formation in C. elegans. So if you have gone through the lectures of the last week, I mean the week 4, so week 4 deals with the vulva formation, the different uh, alleles which are involved, the different pathways which are involved in vulva formation, and what are the mutations that happens. Either it is a gain of function mutation or loss of function mutation, which and what are the phenotypes that ultimately governs those mutations. Okay, and also it dealt with 2-point cross and 3-point cross as well. So this is a question related to the gain of function mutation or loss of function mutations that happens, or the alleles which have such kind of a mutation. So if you have read, if you have gone through the lectures, you would know the meaning of antimorphic, hypermorphic, or complete loss of function. So basically, these are the three conditions: these antimorphic, complete loss of function, and hypermorphic. These are the conditions, and these are the phenotypes. So you have to match that which condition leads to which phenotype. Okay, this you have to match. So I'll give you 2 minutes to answer this question, and then we'll switch the screen, and then we'll see whether you have tried to answer it or not, and otherwise we'll discuss the question. 7-2, I'll switch the screen after 2 minutes. So now I'm switching the screen. Okay. So the question is, we have to match. Just where's my pen? So antimorphic, complete loss of function, and hypermorphic. And the correct answer is first B, second matches to C, and third matches to A. So from here we can say that the correct option is this one. This one is the correct option. Okay, this one. Uh, just a minute. Okay, so this one is your correct option. So now let us try to understand this question. So what is the meaning of antimorphic and hypermorphic? So antimorphic is basically an allele whose gene product acts against the function of the wild type protein. Okay, so if the gene product is acting against the function of the wild type protein, so what will it produce? It produces a dominant negative phenotype. Antimorphic, it produces a dominant negative phenotype. Okay, so what happens because of dominant negative phenotype? There would be loss of function mutation because of antimorphic allele. So here in this case what happens? uh, uh, as we are dealing with lead-16C elegans, which encodes RAS. And RAS is a central signaling molecule in the RAS-MEK pathway, which regulates the vulva development. Now, antimorphic, what does antimorphic allele does? So, antimorphic allele's gene product is against the function of the wild type. So, it will produce a dominant negative phenotype because the mutant protein, it actively antagonizes the normal product. So what will happen? Normally, what should be the phenotype? Normally, in wild type, it should be vulva formation has to be there. But since this is antimorphic allele which acts dominant negative, Negative means it will not form vulva, and dominant means that it is dominant to the wild type. So the product would be vulva less. That means there won't be any vulva formation. So antimorphic allele is there, then there won't be any vulva formation that takes place. Okay. So this antimorphic allele, it basically produces a mutant protein that interferes with the normal, that is the wild type protein. Okay. So this antimorphic, it produces a dominant negative phenotype. So for in this case, as we are talking about lead 60 alleles, what happens for lead 60? This mutant form will lead to block in the signaling, and therefore there won't be any RAS pathway present. And without the RAS pathway, vulva precursor cells will fail to adopt the vulva fate, and therefore the phenotype would be vulvaless. So antimorphic will be vulvaless. Then second is complete loss of function. So when there is complete loss of function, what will happen? It will become lethal. Why it will become lethal when there is complete loss of function? Because see, RAS is essential for many developmental process. It's not just important for vulva formation. Okay, so RAS is important for many other processes also. So a null mutation means, that means a complete loss of function mutation means in lead cysti, it will disrupt the critical pathways which are also necessary for the viability. So what happens, that vulva formation is okay, another thing that is not there, but worms will completely will completely lose RAS pathway and they will die, and therefore the phenotype would be lethal. So when there is complete loss of function, then the phenotype would be lethal. Then hypermorphic. Now what is the meaning of hypermorphic? Hypermorphic is just the opposite of antimorphic. Okay, antimorphic means the morphology is not going to be shown. Hypermorphic means the morphology will be shown more than the normal observed. That means there is gain of function. So such allele that are hypermorphic, it causes an increase in the normal activity of the gene product, which leads to gain of function. So here what happens? Hypermorphic allele makes the gene overactive. So here is excessive RAS signaling. Okay, there is excess of the RAS signaling. There is excess RAS signaling. And what happens because of this? Too much of the RAS, it causes extra cells to adopt the vulva fate, and therefore it will lead to multi-vulva phenotype. MUV means multi-vulva. Okay, so this will lead to multi-vulva phenotype. So phenotype would be MUV. It is a multi-vulva phenotype. So did you understood? So there are two terms, antimorphic and hypermorphic. And antimorphic and hypermorphic are completely opposite of each other. Antimorphic means it is a dominant negative. That means there is no vulva formation. It is vulva-less. And hypermorphic means there will be multi-vulva formation. So it is vulva-less. So this is the correct answer. Just a minute. Fine. So now we'll move to the next question. Our next question is, which of the following was discovered while studying cell lineage in uh, C. elegans? First is, different germ layers give rise to different organs. Genetic regulation in cells gives rise to different cell types. Certain cells are destined to die during embryogenesis, or environment determines the developmental potential of cells. So this is the question. So when the scientists were discovering or studying about the cell lineage in C. elegans, what was the observation that was discovered by them? So what was their conclusion from whatever they have discovered. This is your question. So try to solve this. Think. You have 2 minutes to answer this question and then I'll switch the screen. 1 more minute left. Okay, now switching the screen. Fine. Just a minute. So the question is, which of the following was discovered while studying cell lineage in C. elegans? And the options were: different germ layers give rise to different organs, genetic regulation in cells uh, gives rise to different cell types, certain cells are destined to die during embryogenesis, and finally, last one, environment determines the developmental potential of cells. Okay, so see, if you have studied the C. elegans and its vulva formation, so C. elegans has exactly 959 somatic cells in the adult form. In the adult hermaphrodite, it has 959 somatic cells. Okay, C. elegans. 959 somatic cells. And during the development, exactly 131 cells undergo programmed cell death in a stereotyped manner. So 131 cells during development, 131 cells undergo programmed cell death, that is apoptosis. And this precise cell lineage mapping, it reveals the cell fate that are genetically determined. Okay, so basically this programmed cell death is an important uh, process that occurs during the development of C. elegans, which determines the cell fate, including the death of the organism. So, the correct answer would be certain cells are destined to die during embryogenesis. I hope this is the correct answer. Yes. So, they observed that during embryogenesis, certain cells are destined to die. So, what happens if you have studied, even when we study about the hands of bats and the hands of humans? So, we have digits. Okay, we don't have tissues in between the fingers. So early, through the development process, what happens, it has uh, developed in such a way, it has reformed itself in such a way that the tissues or the cells between the two fingers, the two digits, because of the phenomena of programmed cell death, eventually they died, and then we have individual digits which can move individually. But when you see a bat, then they don't have such kind of a phenotypic feature. So they have a complete like this. Okay, but we humans have individual digits. So the cells which are here, they actually died during evolution with because of apoptosis. So this is what they observed, that during development exactly 131 cells undergo programmed cell death in a stereotype method, and this cell lineage mapping revealed that cell fates are genetically determined. Okay, so if just to show that how this vulva formation happens in C. elegans, you can see this is the anchor cells, and this anchor cell releases uh, inductive signals, and this there are six vulva precursor cells, 1 2 3 4 5 6, and this one, the one which is closest to the anchor cell, which receives the maximum amount of signal, becomes the primary vulva cell, and the one that receives little lesser amount of signal, it becomes the secondary verbal cells. Also, the primary verbal cells also induces the nearby cells through lateral signaling. So this becomes your primary vulva cell, these are secondary vulva cells, and the ones that does not receive any signal, they are the tertiary vulva cells, and they are no, they become hypodermis. So your primary, secondary vulva cells, they form the vulva, and these tertiary cells, they become the hypodermis. So basically these cells, these six cells, they actually form an equivalence group. This is known as equivalence group. Equivalence group means that all the six cells have equal potential to become vulva, but depending upon which cells are receiving the maximum signals, those cells becomes the vulva. Those cells will form the vulva. So this P6th cell, it receives the maximum amount of the signal, which inductive signal, which is coming from the anchor cell, and hence it becomes the primary vulva cell. Whereas these cells which are adjacent to the P6, which are P5 and P7, they receive lesser amount of the inductive signal, which is coming from the anchor cell, and therefore they become the secondary vulva cells. These P5 and P7, they also receive some signal from the P6 through lateral signaling, which goes like this. Okay, this is lateral signaling, which goes like this. And P3, P4 and P8, P3, P4 and P8, which does not receive any inductive signal from the anchor cells, they do not form vulva and they become the hypodermis. So this is just a very basic, you must have studied it, just to give you a basic gist of the vulva formation. And of course there are pathways, RAS, MEK pathway, and EGF pathway, which plays important role in this vulva formation. Okay, fine. Now we'll come to the next question. So ethyl methane sulfonate is commonly used as a dash in genetic screens. Okay, so ethyl methane sulfonate, we know what it is, but what kind of exactly the subset, that you have to tell. Okay, so I'm giving you 2 minutes. After 2 minutes, I'll switch the screen. Meanwhile, you try to answer this question. Okay. One more minute left. 30 seconds more. Okay. So the question is ethyl methane sulfonate is commonly used as a dash in genetic screens. So the correct answer is mutagen. Okay. So what is mutagen? Mutagen is an agent, chemical agent, or any kind of agent that causes mutation. So the agent that causes mutation is known as mutagen. So ethyl methane sulfonate is a chemical reagent or chemical agent that causes mutation. So what does it do is that ethyl methane sulfonate, in short we call it as EMS. EMS. This is basically an alkylating agent. Okay. This is an alkylating agent. So what it does is that it induces point mutation. So ethyl methane sulfonate, it induces point mutations. And because of this point mutation, it mostly causes GC to AT transitions. Because of the point mutations, it causes GC to AT transitions, and therefore it is widely used in genetic screens to create random mutations and then screen for the interesting phenotypes. So basically they are used in forward genetic screenings. Mostly genetic screens, maybe they are used basically in the forward genetic screens. And what is the meaning of forward genetic screens? These forward genetic screens are basically used in C. elegans, in Drosophila, and some plants. So what happens in this forward genetic screens is, the term is forward genetic screen. So what happens is basically random mutations are created and then screened for interesting phenotypes. Okay, this is what forward mutation is. So I hope this question is clear to everybody. Okay. Next question is, hmm, what is the key difference between two-factor and three-factor analysis of genes? Two-factor analysis use two reference alleles, while three-factor analysis use three reference alleles. Second, three-factor analysis can be used to determine the chromosome on which the gene of interest is located on, while two-factor analysis provides information on the nature of the mutation. Third, two-factor analysis can only identify gain of function mutants, while three-factor analysis can distinguish between gain and loss of function mutants. Fourth, Two-factor analysis can be used to determine the chromosome on which the gene of interest is located on, while three-factor analysis identifies the genetic interval on specific chromosome in which the gene of interest is located. Fifth, two-factor analysis can be performed at the F2 generation, while three-factor analysis can be performed at the F3 generation. So these are the 5 points, and carefully read the question. First of all, you should know the basic concept of what is two-factor analysis and what is the meaning of three-factor analysis, because when you know the basic definition of a two-factor and a three-factor analysis, then only you will be able to solve this question. Okay, and uh, then you have to see the difference between them. Okay. Okay, then try to solve this question. I'll give you 3 minutes because there are it's a bit lengthy question, so go through the question carefully, read all the points, and try to understand it. 2 more minutes left. One minute more, then I'll switch the screen and then we'll discuss this question. Okay, so I'm switching the screen now. Let us discuss this question. So the question is, what is the difference between two-factor and three-factor analysis of genes? Okay. So first of all, we should know what is the meaning of two-factor analysis and a three-factor analysis. So two-factor analysis, I'll just do like this. Yes. So first is two-factor analysis. Okay. Two-factor analysis. So basically, these two-factor analysis and three-factor analysis are used in classical genetics approach, especially for C. elegans and for Drosophila. So this two-factor analysis refers to mapping a mutation relative to a single genetic marker. Okay, mapping a mapping a mutation relative to a single genetic marker. So, by following the recombination frequencies between the unknown mutation and a reference mutation, we identify which chromosome the gene of interest lies on. Okay, this is your two-factor analysis. And then when we come to three-factor analysis, three so what happens in three-factor analysis is that the unknown mutation is mapped relative to two known closely linked markers. Okay, here it is mapped relative to a single genetic marker, whereas here it is mapped by two known closely two known closely linked marker. And by looking at the recombination classes, we can figure out whether the mutation lies between those markers or outside of them. So, and this allows the precise localization of a genetic interval within a chromosome. So basically what happens that we make different recombination classes. Whenever we do three-factor analysis, we make different kinds of recombination classes. It is a parental class, recombinant class, and then we figure out whether the mutation it lies between those markers or mutation lies outside of those markers. So this is what we primarily tries to understand. So the question here says, what is the key difference between two-factor and three-factor analysis of genes? So the correct answer is two-factor analysis can be used to determine the chromosome on which the gene of interest is located, while three-factor analysis identifies the genetic interval on a specific chromosome in which the gene of interest is located. So as we just discussed that what is the difference between two-factor analysis and three-factor analysis. So basically with the help of two-factor analysis, you can only determine the chromosome on which the gene of interest is there. But when you are doing a three-factor cross, you will actually get the idea of you will actually get the idea of the genetic distance or the genetic interval on that particular specific chromosome. So this is what is the basic difference between a two-factor analysis and a three-factor analysis. Okay, so let us come to the next question. It is again a very little lengthy question to understand. The question says, on a chromosome we situate three mutant alleles of three different genes A, B and C, okay, to study the recombination frequencies among them. Fine. We observe that the recombination frequency between A and B is 26%, while the recombination frequency between B and C is 10%. Fine. Additionally, the recombination frequency between A and C is 18%. Okay. Which of the following inferences is correct? The allele for C is between A and B and is closer to A. Second, the allele for A is between C and B and is closer to C. Third, the allele for B is between A and C and is closer to C. Fourth, the allele for B is between A and C and is closer to A. Fifth, the allele for C is between A and B and is closer to B. And sixth, the allele for A is between C and B and is closer to B. So this is the question that you have to solve. I'll give you 3 minutes because it might take some time for you to understand and solve this question. So what you have to do is, whatever information is given to you, try to make a diagram out of it and then try to solve it. One hint then I can give you, that it is an example of a three factor cross. It is a three factor analysis. This question is based on three factor analysis, and just now we have discussed that three factor analysis, it identifies the genetic interval on a specific chromosome in which the genes are located. That means by looking at the classes, we can figure out whether uh, we can figure out the precise location of the genetic interval. Okay, so this is the hint that I am giving you right now. It is 7:38. I'll switch the screen after 3 minutes. I'll see whether you are able to answer it or not, and then we'll discuss. It's a very interesting question, and it will clear a lot many concepts about three factor cross also. Okay, so try to solve this question. Try to uh, the way to solve these questions is to make a diagram. Okay, so try to make a diagram. Whatever information is given, write that information on your copy, and then whatever numbers are given, 26%, 10%, 18%, accordingly try to make the figure. Okay, and then it will be easy. It's easy, but it's also conceptual. 2 more minutes left. Try to make the figure, because that will only help you to solve this question. Okay, make the figure and then solve it. One more minute left. Okay, now I'm switching the screen. Fine. Let's see if you have anything. Okay, so now let us try to understand this question. It is a question regarding 3-point test cross. So, whenever there is a question which is about 3-point test cross, the recombination frequency which is given between the two genes or the two alleles, this is basically the distance between them. Okay, so the recombination frequency is basically the distance between the two alleles. Suppose if the recombination frequency is between A and B is given to you as 26%, which means that this is the distance between them. This is 26 centimorgan distance is between A and B. Now let us try to make the figure and solve it. We'll solve it. It's very easy. There are three mutant alleles A, B, and C. Okay, just I'll put it here. Yeah. So there are three mutant alleles A, B, and C. Now the recombination frequency between A and B is 26%. That means the distance between them is 26 centimorgan. Okay. The recombination frequency between B and C is 10%. Okay. And the recombination frequency between A and C is 18%. Here it is 18%. So from this, what can we say? We can say that the distance between A and B is the most. Okay. So suppose if I keep C here, A here, and B here. So between B and C, it is 10%. Between A and C, it is 18%. And between A and B, it is 26%. I mean, the 26 centimorgan, you can say. So, does it make sense? Yes. See, plus minus 2, that much small error is expected. So, you can see that this equals to 28 centimorgan, and they have also given 26 centimorgan. This plus minus 2 of the error is expected. So, the map would be like this, that there is A, C and B, and because the distance between C and B is 10 centimorgan and the distance between A and C is 18 centimorgan, we can say that C is nearer to B as compared to A. So this is the map. So now let us try to see the allele for C is between A and B and is closer to A. No, this is wrong because it is closer to B. The allele for A is in between C and B. It is again wrong. The allele for B is between A and C. It is again wrong. The allele for B is between A and C. Again wrong. The allele for C is between A and B. C is between A and B. And it is closer to B. So this looks like the correct option. Let us see whether it is correct or not. Yes. So this is the correct option. The allele for C is between A and B and it is closer to B. Okay. So the for solving these kinds of questions, what you have to do is that this recombination frequency is actually telling the distance between the two alleles. Okay. So the recombination frequency between A and B is 26%. That means the distance between the allele A and B is 26 centimorgan. The distance between B and C is 10 centimorgan. The distance between A and C is 18 centimorgan. So according to the distance that is given, you can try to make a map. And then you can easily solve this question. So 3-point test cross, that is the beauty of 3-point test cross, that it can tell you the precise location to a genetic interval. It can tell you that which gene is in between. So basically, whenever you are doing questions which are related to 3-point test cross, they they actually ask you such questions. Okay, they ask you the questions, which gene is in between, the distance is given, make a map. So these are some regular kinds of questions which comes whenever we are dealing with the 3-point test cross questions. So I hope this is clear to you. Now let us move to the next question. Okay, just a minute. Hmm. Next question is, What are the major advantages of suppressor screens? Okay, what are the major advantages of suppressor screens? First, it allows for screening of lethal phenotypes of mutant alleles. Second, it provides insights on gene to gene interactions. Third, it allows heterozygous mutants to be used to determine the gene function. Fourth, it allows for screening of gain of function mutants. And fifth, it allows for identification of function of new mutant alleles in other genes. So these are the five options. Okay. I'm giving you 2 minutes. Try to answer this question and then we'll discuss it. One more minute left for you to answer the question. Okay, now we'll switch the screen. Okay. So the question is, what are the major advantages of suppressor genes? Uh, yes. So, it allows for screening of lethal phenotypes of mutant alleles. It provides insight on gene to gene interactions. It allows heterozygous mutants to be used to determine gene function. It allows for screening of gain of function mutants. It allows for identification of function of new mutants in um, another genes. So before answering this question, we should first know what is the meaning of suppressor screens. So suppressor screen is basically a genetic screen in which researcher starts with a known mutant. Okay, known mutant means a mutant which has a clear phenotype, and then look for second side mutations that suppresses the original mutant phenotype. Okay, so basically they look for side mutations that suppresses or reduce or eliminate the original mutant phenotype. So basically the primary mutation is a but is something which is a strong which has a strong and a visible phenotype, and suppressor mutation is the second mutation in the same gene or in a different gene also, that restores the function of the normal, that restores your normal function, and it can restore the normal function either partially or it can restore the normal function fully also. So the goal is basically to uncover the functional relationship between genes and the pathways. That is the goal. So, for example, in C. elegans also, we suppressor screen is usually is widely studied. So, what we can say, summarize, that suppressor screens are the genetic strategies which helps to identify the secondary mutations, and these secondary mutations are the mutations that rescue the phenotype of the primary mutation, thereby it uh, reveal the gene to gene interaction or gene to pathway interactions. Okay, and these suppressor screens are powerful because they look for secondary mutation that suppresses the phenotype of the primary mutation. Because see, suppressor screen, this is important, that suppressor screens, it basically looks for secondary mutations that suppress the phenotype of the primary mutation. Okay, this is important. Now let us come to the options. First is allows for screening of lethal phenotypes of mutant alleles. Is this correct? Yes. Because if the mutation is lethal, you cannot study it. But because of this suppressor screens, if a second site suppressor, it reduces the viability, rescues the viability, you can analyze the underlying pathway. So yes, this is the correct one. Then provides insights on gene to gene interaction. Again, it is correct because suppression often indicates that the two gene act in the same pathway, or they play parallel pathways role, or they play antagonist role. So whatever kind of role is there, that can be analyzed with the help of gene to gene interactions, which helps to build overall a network. So then this is also correct. Then allows for allows heterozygous mutants to be used to determine gene function? No, this is not correct. Why? Because suppressor mutations frequently occur in genes which are different from the original one. So this way, yes. Uh, Basically, this is not a specific advantage of the uh, suppressor screens. Here, see, whenever you are dealing with such questions, you should see the options. Here, you have to click or select the advantages of the suppressor screens. This is not specifically an advantage of suppressor screens. This is a wrong statement. Then it allows for screening of gain of function mutants. No, that's usually handled with specific dominant or gain of functional mutational studies, not with suppressor screens. So this is again wrong. Then fifth is allows for identification of function of new mutant alleles in other genes. Yes, this is correct because suppressor mutations, it frequently occur in genes which are different from the original one. Therefore, you can discover the new players which are in the same biological process. So this is the correct statement. So you have three correct statements, 1, 2 and 5. Let us see whether it is correct or not. Yes. So the accepted answers are allows for screening of lethal phenotypes of mutant alleles. It provides insights on gene to gene interactions, and it allows for identification of function of new mutant alleles in other genes. These are the functions of or the advantages of suppressor screens. So I hope what is the meaning of suppressor screen, that is clear to all of you. Fine. Let us move to the next question. Okay. The question is, two genes codes for enzyme A and B. A loss of function mutation in gene B gives the same phenotype as the loss of function mutation in genes A and B. What are the possible inferences you can make about the pathway? A. Enzymes A and B are part of sequential substrate pathway where enzyme A acts before enzyme B does. 2. Enzyme A and B are part of sequential substrate pathway where enzyme B acts before enzyme A does. Third, enzyme B is epistatic to A. Fourth, enzyme A and B are part of regulatory pathway where enzyme B activates enzyme A. Fifth, enzyme A is epistatic to B. And sixth, enzyme A and B are part of regulatory pathway where enzyme A activates enzyme B. So try to answer this question. You have 2 minutes and then I'll switch the screen and then we'll discuss it. 1 more minute left. Okay. So the question says, Two genes code for enzyme A and B. A loss of function mutation in gene B gives the same phenotype as loss of function mutation in genes A and B. So what does it mean? So loss of function in gene B gives a, suppose if gives a phenotype X, okay? When there is a loss of function in gene B alone, when there is a loss of function in gene B alone, it gives a phenotype X. When there is a loss of function in both A and B, then also it gives the same phenotype X. What does it mean? This means that mutation in B, it masks the effect of mutation in A. Okay, so from here you can say that mutation in B, it actually masks the effect in mutation A, which is an example of what? This is an example of epistasis. Okay, then sequential pathway logic. What is sequential pathway logic? That just a minute. See, if enzyme A acts before the enzyme B, then without A, substrate can't be converted to the intermediate. Without B, intermediate can't be converted to the product. So this means that B is epistatic to A. So this enzyme B is epistatic to A. This we can get to know. Then what happens? What is happening? Actually, B is forming A, which is forming the product. So if B is lost, the pathway stops before A even acts, losing A as well as changes it nothings. So B acts before A and B is epistatic to A. So there is one option where it says that enzyme B, yes, second one. I think second is also correct. Yes, because B is acting upstream of A and A activates B. How A activates B? Because it's a regulatory model. All are compatible with the genetic results. So the answer would be this, and sixth. So first logic is your epistatic logic, where only B is also giving the same phenotype as A and B both. This means that B is masking the effect of A, which means that B is epistatic to A. Second is your sequential pathway uh, logic, where the order is first B is converted to A and A is converted to product, because B is epistatic to A. Therefore, whatever B makes, it is directly converted to the product, which means B is coming before to A, and A basically activates B. So this is the correct answer. Yes. Now next question is, C. elegans gene X has three. C. elegans gene X has three single nucleotide polymorphism SNPs A, B, and C. The strain homozygous for SNP A is crossed with the strain homozygous for SNP B, and the F1 progeny are crossed with the strain homozygous for SNP C. If a male is randomly picked from the pool of F2 progeny and crossed with the strain homozygous for C, what is the chance of progeny having at least one A allele? First is 100%, second is 75%, third is 50%, fourth is 25%, and fifth is 0%. Okay, so I think I'll directly go with the question and I'll try to answer it. Okay, because this is a little not typical, but I think it's better that I should start solving it. Hmm. What the question says, C. elegans genes has X has three single nucleotide polymorphism A, B, and C. The strain homozygous for SNP A. So homozygous for SNP A means it would be AA. Okay. AA is crossed with a strain homozygous for SNP-B. So AA is crossed with B. This is the first thing that they are saying. So what is formed? AB, 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 AB. Okay. This is the F1 progeny. This is this is the F1 progeny. So what they told? They said that the strain homozygous for SNPA. Homozygous means that both the alleles are same. So the strain homozygous for SNPA, which is AA, is crossed with the strain homozygous for SNPB, which is BB, and the F1 progeny. What is the result of F1 progeny? It is AB. The F1 progeny are crossed with the strain homozygous for SNPC. So this means this is the F1 progeny. This is crossed with CC. So this is C and this is A and B. Okay. What they are saying is that the F1 progeny, this is the F1 progeny, is crossed with a strain homozygous for SNPC, which means CC. Homozygous means both the alleles are same. So what is the result? It will form AC, AC, BC and BC. This means 50% it forms AC and 50% it forms BC. So I hope up till here it is clear. Now, the question comes, if a male is randomly picked from the pool of F2 progeny, and this is the F2 progeny, this was the F1 progeny, and this is the is randomly picked from the F2 progeny and crossed with the strain homozygous for CC, what is the chance of progeny having at least one allele if a male randomly picked from the pool of F2 progeny? So suppose if the male is randomly picked from a pool of F2 progeny, male can be either AC or BC. Okay, if the male is BC, is there any chance of having A allele? No, because BC has no A and because it is crossed with homozygous C. So from here there is no chance, but even then we are going to solve it. What is the chance of progeny having at least one A allele? So let us see. Suppose if the male chosen has AC alleles. So suppose the male has case A AC allele in the F2 progeny. Okay. It is crossed with the strain homozygous for C. This means it is crossed with CC. This is AC. Okay. AC. AC, AC, CC, CC. So from here, what is the chance of progeny having at least one A allele? So here the chance of progeny having at least one A allele would be what? How much? It would be it would be chance of male having AC into the chance of progeny inheriting A. Okay. So this would be chance of male having AC is 50%, which is 0.5, and chance of the progeny inheriting A is also 50%, which is 0.5. So which becomes 0.25, which is 25%. And if the male has BC as the allele, then the chance is 0 because here there is no A, and because it is crossed with CC, so here also it is null. So the correct answer would be 25%. So I hope this question is clear to everybody. You need to understand the last thing. See, the question is saying what is the chance if the male is randomly picked from the pool of F2 progeny and crossed with a strain of homozygous C? What is the chance of progeny having at least one A allele? So here you have to do multiplication. Why? Because first the probability of picking AC allele and then the probability of having A from that AC allele crossed with CC. Okay, so therefore it would be 0.5, 50% here into 0.5, 50% here, which is 0.25. This is the probability. So I hope this question is clear. Okay, any problem in this question, you can ask me. So see, from here, we have to pick randomly. So randomly, if a male is chosen, it can either have allele AC or it can have BC allele. If it has BC allele, and according to the question, when it is crossed with CC, what will be the chance of A? Zero. Because there is no A allele here, right? Here, if suppose AC is chosen and then again when it is crossed with CC, so there are two probabilities. We have to multiply both the probabilities. First, the probability of choosing of AC from this lot and then the probability of having A from this lot. So this is 50%, there's 50% which is 0.25, 0.5, 0.5, 0.25%. It is 25%. So I hope this question is clear. Now next question is, what is the ground state of PNP cells in an uninduced induction? Primary, secondary or tertiary? What can be the answer? See, I'll explain you. This is an easy question. Oh, sorry. Sorry, is there. Fine. Doesn't matter. See, we have already discussed this with the help of a diagram which was here, I think, somewhere. Where is that diagram? Yeah, this was the diagram. You see, this is the primary fate, this is the secondary fate, and this is the tertiary fate. So primary fate is, these are the vulval precursor cells and they form an equivalence group. What is the meaning of equivalence group? That all these vulval precursor cells have the equal potential of forming vulva. It all depends upon how much inductive signals they are receiving from the anchor cell. This sixth one, P6P, it becomes primary because it receives the maximum amount of inductive signal. These two cells receive lesser amount of inductive signal from the anchor cell and some of the signals they receive from the P6 through lateral signaling. Therefore, they become the secondary cells. And P3, P4 and P8, they do not receive any inductive signal from the anchor cells and therefore they become the tertiary because they are not getting induced. Okay, so basically anchor cells are inducing these primaries, these vulval precursor cells. This is an example of induction. Okay, so there it is primary, secondary and tertiary. Now the question here says, what is the ground state of PNP cells in an uninduced condition? Uninduced condition means a condition where they do not receive any inductive signals. Uninduced means they are not induced and they are induced by the inductive signals which are coming from the anchor cells. So in the uninduced condition, they will be tertiary because they are not getting induced. Maximum induction will lead to primary cells. Okay, here they have the maximum induction. Little lesser, they become the secondary cell. Completely not receiving any of the induction, then they become the secondary. Okay, this is the default state. This is the default or the ground state. This is the or the ground state. Okay, so I hope this is clear to everybody. Now let us come to the last question. Cosmids, Back, and Yak are used to determine the dash map of the chromosomes of different organisms. Okay, so these are basically cosmids, Back, and Yak. These are basically your plasmids. Back is bacterial artificial chromosome, Yak is yeast artificial chromosome. These are bigger plasmids. So these are used to determine the dash map of the chromosomes of different organisms. So I'm giving you 2 minutes and then I'll switch the screen. Is there any other question also? No. Fine. Hmm. Right now it is 8:18. I will switch the screen after 2 minutes. That is at 8:20. Okay. Think what kind of maps are present or used in genetics. If you know that, it will be little easier for you to answer the question. One more minute left, and then I will switch the screen. Okay. Let us try to solve this question. Okay. Hmm. Cosmids, Back, Yak are used to determine the physical map of chromosome of different organisms. Okay, physical map. The correct answer is physical map. So now you have to understand that there are basically three types of maps which are used in genetics. So in genetic studies, there are three types of maps which are used. So first is the genetic map, which we called as the linkage maps. Okay, genetic map, which is known as the linkage map. Second is the physical map, and third is the cytogenetic map. Cytogenetic map. Okay. Now, genetic map is basically based on recombination frequencies between markers, and the units are centimorgans. So the genetic map that we basically use to solve questions like 2 point test cross or 3 point test cross. So basically, these are based on recombination frequencies. Okay, these are based on recombination frequencies. Whereas physical maps, these are based on the actual DNA sequence or clone fragments, and they basically tell the real physical distance between the two genes. Okay, these are the recombination frequencies. That means how much the gene has undergone recombination. So we have parental types and recombinant types, but physical map is telling you the real physical distance between the genes. So this physical map, it tells the real physical distance between the genes. Whereas cytogenic map, this is basically based on chromosome ka banding pattern. Okay, this is based on chromosome banding pattern. Now, these cosmids, BAC, and YAC, these are basically large cloning vectors. I told you these are large insert cloning vectors, and basically these allow the researchers to maintain long stretches of DNA inside the clone. And by arranging overlapping clones, which we called as contigs, you can actually reconstruct the whole genome. We reconstruct the whole sequence. And why they determine the physical map? Because these vector contain the actual pieces of DNA which are aligned by overlap, and then they reveal the true physical order and the true spacing of the genes. Okay, so they reveal the true physical order of the genes. And this is different from a genetic map. Why? Because genetic map only gives the relative distance based on recombination, but this is giving you the actual physical distance. Okay, so therefore cosmids, BAC, and YACs, they are used to determine the physical map. The actual, the real physical distance between the two genes is determined by cosmids, BACs, and YACs. So remember, this is important whenever you're studying about physical map, that physical maps are based on actual DNA sequence or clone fragments. So these are basically large cosmids, BACs, and YACs. These are large cloning vectors. I mean, they can have the cloning of large inserts. Okay, so I hope they have the cloning of large inserts. So I hope this is clear to you. Hello. Okay, so just to out of all the questions, I think major things that you should remember are uh, yeah. So antimorphic, what is the meaning of antimorphic? Antimorphic is dominant negative. So when I'm talking about dominant negative, what does it mean? It means that this kind of allele, basically it acts against the function of the wild type protein. And because it acts against the function of the wild type protein, it interferes with the normal functioning or the normal wild type protein. Therefore, it is dominant negative. It is loss of function mutation. Whereas hypermorphic, what is hypermorphic? Hyper means more. Morphic means morphology. Antimorphic means ki morphology is not going to be shown. Hypermorphic means more morphology will be shown. That means there is gain of function mutation that causes increase in the normal activity of the gene. Okay, then we were discussing about the vulva formation. I already told you that these are the equivalence group. Anchor cell releases the inductive signals, and the one which is closest to the anchor cell receives the maximum amount of signal, and it becomes the primary vulva cells. The second, which are adjacent to the primary vulva cells, become the secondary vulva cells because they receive lateral signaling, lesser signaling as compared to the primary vulva cells. And P3, P4, P8, they are not receiving any signaling at all, so they become tertiary, which ultimately becomes the hypodermis. Then ethyl methane sulfolate, it is an alkylating agent. It is a mutagen which causes GC to AT transitions, and this is used basically for forward genetic screens. What is the meaning of forward genetic screens? Forward genetic screens may random mutations are created, and then they are screened for the interesting phenotypes. Then two factor test cross and 3-point test cross. I told you what is the difference between a 2-point test cross and a 3-point test cross. In 2-point test cross, what happens? It is basically used to map a mutation relative to a single genetic marker. Important is that there is only a single genetic marker which is used. Whereas in the case of 3-point test cross or three factor analysis, what happens that unknown mutation is mapped relative to two known closely linked markers. Okay, so basically by looking at the recombination classes, we can figure out whether the mutation lies between those markers or it lies outside those markers. Okay, then we deal how to solve these kinds of 3-point questions. Remember, recombination frequency is actually the distance, I mean the distance between the two genes, the genetic distance between the two genes, which is expressed in centimorgans. Suppose if it is written recombination frequency between A and B is 26, this means the genetic distance between A and B is 26 centimorgan. Recombination frequency between B and C is 10%, which means the genetic distance between B and C is 10 centimorgan. Recombination frequency between A and C is 18%, which means the genetic distance between A and C is 18 centimorgan. This is what you have to remember. Then other important thing was the mapping, genetic mapping, physical mapping, and cytogenetic mapping. Genetic mapping basically is based on recombination frequency. The linkage, we also call it as linkage mapping, generally used in C. elegans and Drosophila to study 2-point test cross and 3-point test cross. Physical mapping, it tells the real physical distance between the genes. And cytogenetic mapping is basically, it tells based on the chromosome ka banding pattern. Cytogenetic mapping is based on chromosome banding pattern. So with this, we have completed the questions which are related to the week 4 class. In the next class, we'll deal with the questions for from week 5 classes. So I hope it was clear to you. We'll meet in the next class the same time, Tuesday 7 to 9 p.m. So bye.